Here we have part 2. That's supposed to be a 2. That was a little wavy there. Page 64. And we're still doing cytology, the life of the cells, individually and collectively. And so we're looking at membrane right now. And I have some annotation here that, and there's going to be a paper that we're going to look at pathology of the above, the membrane right here. There's going to be the potential for pathology. Out here we have a capillary, usually a whole set of capillaries that are providing nutrients from the bloodstream to the cell here for the cell's nourishment. Also, the cell is metabolizing these nutrients, and we're getting waste products as a result of that. So oxygen in, CO2 out, substrates in, and byproducts, possibly amino groups or urea out. And so we need the capillaries close at hand, and there's a matrix between the capillary and the cell that is of great interest to us. We call that the endothelial matrix. That's where nutrients and waste products are exchanged between cells and the bloodstream. Okay, and so we have this issue going on here that I'm referencing, a pathology of the above with reference to metabolic syndrome results in advanced glycosylated end products. So please see cardiovascular diapathology review paper, and we're going to be considering a review paper in which we look at the capillaries coming close by here and how the capillary walls and the basement membrane actually thicken in the case of metabolic syndrome when people have a very sugary diet. Sugary could also be a starchy diet in which the starches are broken down into sugars. Right, so in that the glycosylation, you can see already here that on the cell itself, there's species-specific methylations and other types of how are they referencing it here as the glycoproteins. So these are the sugar, how shall I say, or carbohydrate signals on the outside here. And there's going to be the buildup of dietary sugars in the capillaries that are going to change the nature of the matrix that's between the capillaries and the cells. So we have to consider that, especially this is true of people who are diabetics, type 1 or type 2, and they endure issues with regard to challenges to perfusion. We'll get into that because that's our next topic. Okay, so the glycocalyx will be part of that little program right there, the cells in terms of their surface. Here we have some cells that are being held together, and they're being supported by a basement membrane. And we have an apical surface here where we have a lumen out here. It could be the inside of the intestine, for instance. And down here we have the wall of perhaps the intestine. And then we have these cells right here, and they're being held together. And you'll notice they're being held together in different ways. One such way is illustrated right here with kind of a continuous type of bonding that occurs between the membranes of the cell. And that bonding is referenced here as tight junctions. Okay, so you have to think to yourself, this is like the way you hold your six-pack of Coca-Cola together or something like that, in which there is a plastic unit, or there's a continuous type of bonding that goes all the way around each of these things and holds them in place. This next one is interesting. It's called a desmosome. That's number 2. The first one was tight junction. The second one here is desmosome. The third one here is gap junction. But the desmosome is particularly complex in that there's all of these features here. There's this little plate on each side. There's the part that fits together, sort of interdigitates together. There's the keratin fibers, which are actually, keratin is what makes your hair, for instance. And so there's these little fibers that go in and sort of maintain the pitch of each cell with regard to the other cell. Okay, and notice that there's a little space that's maintained. So since there's no blood vessels close at hand, and the nutrients have to come from underneath the basement membrane. That means there can be capillaries here, and that nutrients can filter up through that space. Do you see how nice that is? So we need to have desmosomes for certain types of tissues in order to create that space between the cells. Then we have these things over here. The next one, gap junction. A gap junction is particularly useful in a tissue, particularly in the heart, for instance, where we have a muscle that contracts in the heart, and it actually activates the muscle immediately next door to contract next. It creates a wave. If you've ever been in a stadium, a large stadium, in which one person stands up and sort of taps the next person, and they stand up along with them, and that person taps the next person, and so on, then we get a wave of actions. And in the case of a heart, it wants to be a wave of depolarization and contraction so that we move the liquid within a chamber of the heart. It's very difficult to move liquid through just pure muscle contraction, so you have to do it in this very organized wave-like manner, and that's what gap junctions are for. Gap junctions. Okay, so there's that. And then over here we're going to have a brief discussion of diffusion. Diffusion is discussed here. We're looking at page 68 here. There's active and passive processes of diffusion, and we're immediately here, first of all, going to look at the passive processes of diffusion. Okay, and diffusion is predicated on what's referred to as concentration gradients. Okay, there's different ways of creating a gradient. One way is you have, for instance, I'm imagining that I'm dripping something like ink into this container. This looks like a solid, actually, but let's imagine that there are some drops of dark blue ink or something like that being dripped into a container of water. Here there's a high concentration of the ink molecules, and over here there's a high concentration of the water molecules, and so they're colliding with each other. Why are they colliding with each other? What is, what causes any of these liquid molecules in a liquid format to be colliding? And the reason for that is they're not at absolute zero. And so you'll have to take my word for it. You can look at Brownian motion under the microscope and see with certain things that can be, if you put a floor on it and if you look carefully, you can actually, at high enough magnification, see movement of certain types of molecules. But at any rate, there is movement here, and there is collision between the molecules. It's liquid, so they're sort of like bumping into each other and then moving away through that liquid medium. And there's a lot of bumping going on here. And as they begin to bump into each other, they spread out a little bit. There are fewer and fewer and fewer collisions as we move this way, so we're moving from a high number of collisions here to a lower number of collisions here. That is considered a diffusion gradient. It's a gradient based on concentration of particular molecules. Okay, and so we're getting movement here, and we're getting, mixing of the water molecules with the ink molecules. If we go away and come back after a long enough time, we notice that everything has been dispersed equally and homogeneously throughout the system, which is to say there's no more gradients. There's still going to be collisions, but there's as many collisions over here as there are over here. Okay, so that's the idea here, is that there's the same number of water molecules here as here, the same number of ink molecules here as here. So the number of collisions have more or less, we call it equilibrated. Now, bear in mind that I've already said once in this course that equilibrium equals death. Right, and so we don't have homogeneous systems within the body. So we're just looking at this as a chemical system. This is something we can accomplish in a beaker with water and ink. Water as a solvent, and anything that dissolves in water as a solute. Okay, so it's possible to have this system at rest finally, and that is equilibrium.